Hello everyone and welcome to this new episode of Learn Stress Free. I'm your host, Jeff Rodrigue. My goal with this podcast is to make scientific knowledge accessible to the public in a way that's fun and easy to understand for everyone, even your 5-year-old cousin. All thanks to my expert guests who conduct research in academia. In today's episode, we're going to talk about RNA. Our guest will explain what RNA is and why it's so important to learn about it. Drawing on her expertise and extensive scientific research, she will debunk some common misconceptions and misinformation surrounding RNA and answer the following questions, among others. What is RNA? Why is it so important to conduct research on RNA? And what are the benefits for society of scientific advances in this field? Is RNA and DNA the same thing? How long has the scientific community been interested in RNA? Here's a hint, it's been since well before the COVID-19 vaccines. In this episode, thanks to science, you get answers to these questions and more. Without further ado, let's learn stress-free with Karine Choquet from the University of Sherbrooke. Hello everyone, welcome to this episode of Learn Stress-Free. Today we're going to talk about RNA to understand what it really is by addressing misinformation and highlighting the many benefits of scientific advances related to RNA for all of us as part of our society. But first of all, as you know, I love science, but I'm more interested in the people who do the science. So let's us meet our guest, which is Karine Choquet, professor at University of Sherbrooke. Hi, Karine.
Hi.
So thank you very much for joining us today. Before we dive into science, we would like to know who are you? And since no one knows you better than yourself, I'll let you introduce yourself to our audience.
Sure. Well, first, thanks for having me. So as was mentioned, I'm an assistant professor at University of Sherbrooke and also a researcher at the Research Center on Aging in Sherbrooke as well. And so I've had a longstanding passion for genetics that started when I was a teenager, and I was a huge bookworm at the time. And I came across a novel on a geneticist that was studying genetic traits in twins. And it was a mystery novel. And the study of genetics helped the character to solve a mystery. And around the same time, I had my first introduction to genetics in high school biology. And those two things together really came together to make me want to pursue a career in genetics. And so fast forward to the end of high school, I went to do an undergraduate degree in biochemistry and molecular medicine at Université de Montréal. And this was followed by a master's and transitioning into a Ph.D. in human genetics at McGill University. And so during my undergrad and Ph.D., I really focused on the genetics of rare neurological diseases, some of which are a little bit more frequent in the French-Canadian population. And one of these diseases had to do with defects in RNA, which we're going to talk more about in a few minutes. And so this made me want to learn more about how to study RNA. So I went to Harvard University to pursue a postdoctoral fellowship in RNA biology. And then following that, I came back to establish my independent research group at University of Sherbrooke, where I sort of combine these two aspects of genetics and RNA biology.
So very, very interesting. Thank you very much. So RNA, so you study RNA, but RNA kind of has a bad reputation, sadly, on social medias with the development of the RNA vaccines, such as the one for COVID. But without getting into specifics of the vaccine themselves, I went on your research lab's website, and it's very interesting, but there are a lot of words I didn't understand at all that I needed to Google, and not just because I'm French. And so if you want to play Scrabble and score big, there's a lot of points to go get there. But to start from the beginning, basically, can you explain to our audience in terms that even I can understand what exactly is RNA, what does it actually do, and why is it important to understand it?
Yeah. So first, I'd like to specify that RNA is not recent. It's something that's been around for thousands of millions of years. So it's present in all living organisms. And we do research on RNA. We've been doing research on RNA for decades because of that reason. It's really essential for the life on Earth, for example. And so to explain what RNA is, I'll start from DNA, which is a slightly different molecule. And we've often heard of DNA as being where our genetic information is stored. So we can see DNA as a giant instruction manual that's separated into different paragraphs, and those paragraphs are genes. Now, genes contain the information to make proteins. And you might have heard of proteins in the context of nutrition, but really proteins are like the building blocks of our cells. So if DNA is the instruction manual, the proteins are sort of the workers that make everything happen in our cells, from digesting nutrients to protecting us against microbes or viruses. And so DNA contains the information necessary to make proteins. But where it gets more complicated is that, as I mentioned, DNA is this huge instruction manual, and it's actually physically separated from the place where protein is made. So its DNA is in the part of the cell that we call the nucleus, and there's like a big gate around the nucleus, whereas proteins are made in another part of the cell. And so it's as if DNA was sort of stuck inside this area with the fence, and it's too big to get across to get information to proteins or to the place where proteins are made. And so it needs a message. And RNA, in this context, is the message. That's why we call it messenger RNA. So RNA is copies of just one paragraph of DNA at a time. So it's small enough, it can get across the fence and bring that same information that's contained in DNA to the place where proteins are made. So we say that one messenger RNA contains the information to make one protein. And so RNA is like a temporary copy of DNA. And I mentioned one reason why we might want a temporary copy, so that it can get across that big fence. But the other reason is that DNA is precious, and we don't want to damage it by sort of soliciting it too much. Whereas RNA is very temporary. It can be replaced easily. And so we use it in this context as the message between DNA and protein.
You mentioned RNA is part of, basically it's been existing since forever, and it's part of every human, not only human, but every living being, every living organism. So what does it do in our systems, basically, that RNA versus the DNA? Because the DNA is more like central, it's stable, and the RNA is more temporary.
Yeah. So as I mentioned, the RNA is like the message. So like another analogy would be, for example, like if you want to, let's say, build a house, you could say that the house is like the protein, and you might have, many different plans for a house that you could choose from. But you might, as an architect, you might not want to bring that huge book of different options for houses to, where you're building the house. You'll choose just one. And so the RNA could be just that one plan that you choose. And so going back to the idea that DNA needs to be protected, part of that is because we have DNA present in only 2 copies in each cell. So generally, one copy comes from one parent and the other from the other parent. Your DNA has to last a really long time, so we don't want to damage it. By contrast, mRNA is much more dynamic. And so you could have, like, let's say, the RNA copy from one gene be present in only one copy, and another one, if it's needed a lot, will be present in thousands of copies. And this changes through time and as a function of, let's say, environmental conditions. So if it's really hot outside, you might make more of one RNA that helps you to deal with that heat. Similarly, the from all the possible genes that are present in the big instruction manual that is the DNA, not all of them are needed in every organ, for example. So there's only some of those genes that will be copied into mRNA in the brain, whereas there's another set of genes that will be copied into mRNA in the liver. So RNA, not only is it the message that allows to transmit the information from the instruction manual to the workers, so DNA to protein, but it also allows our system to be really flexible in terms of which messages it wants to transmit.
That's why you say basically RNA is more temporary, because you mentioned RNA can be a signal that is sent if it's warm. So our system needs to react to this to cool us off. But you don't want that message to stick around, because eventually if it's not warm anymore, you don't want your system to keep on doing those functions. It needs other functions. So that's why RNA is basically more not time and context specific. Is that, if I may?
Yeah, exactly. Yeah.
So basically, the RNA, so what you do with your research also, it's useful to basically understand what part of the message needs to be sent, where and when, and how to send this message to really understand it. Am I close?
Yeah. So I guess specifically with regards to my research, we go a little bit deeper than what I've talked about so far. So I've told you about how DNA needs to be copied into mRNA. But when the paragraph from DNA is first copied into RNA, it contains words that are, really clear, that contain instructions to make proteins. But each of those words is separated by more complex sections of code that may appear less understandable to the naked eye. So an analogy would be like one of those hidden word puzzles where you have a number of different characters and you have to find words within them. And so that's what RNA looks like when it's first copied into DNA. And so there's a process called splicing, which is what we study in my lab, where those sections of code that is more complex have to be removed, and the normal words are pasted back together to form a real sentence. So for example, if we use the analogy of sentences, it could be like, it is really hot outside. Now, why have that complex information in between the normal words? Well, it helps to increase the flexibility of which sentences we can make. So perhaps there's also other words in that big jumble of words or of letters that can be used to make different sentences. So I talked about one, which is, it's really hot outside, but perhaps there's an alternative word that could be used to make a slightly different sentence. It's really nice outside. And you could also combine them and say it's really hot and nice outside. And so this is just to illustrate that from one paragraph in the DNA, you can make many different mRNAs that are slightly different, and that will make proteins or workers that have slightly different functions. And so again, one of these sentences could be used in the liver, and another one could be used in the brain, for example.
And understanding those words and how you arrange those sequence, I know it's not words per se, but understanding the sequence and how it works, I guess, does it also help to know, is the body sending the right messages?
Yeah, and that's sort of the other aspect of my research and that of many other researchers, is that this process, splicing, is known to go wrong or to be defective in a lot of human diseases, and it also becomes less and less accurate as we age. And we don't really know how or why this happens. And so if we can understand how splicing happens, so how those words are put together in the correct order to form a sentence that makes sense, this can help us better understand how and why it goes wrong in different diseases, and eventually to use that to prevent or treat these diseases.
So basically, by understanding the message that should be versus the message that is actually sent in the body, because I guess when the body sends the wrong message, or the message is missing or is wrongfully interpreted, that's how you have body dysfunctions. And part of your research and part of why it's important to understand RNA is to make sure that we can rearrange the word and make sure that the communication inside the body communicates the right information to basically correct this dysfunction and to solve it.
Yeah. So, like as an example, in certain cancers, the cells start to grow uncontrollably, so to become cancer cells, basically, because of maybe skipping a word or keeping part of that code that should be removed, but it's still there. And so all of that can cause problems down the road that can lead to diseases.
And you mentioned also aging, which, well, we can't escape, right? All of us, we're all moving towards every second we age. So how can it apply to aging, and how can it help the process of aging maybe in a more healthy way to understand RNA?
Yeah, so that's a big focus of my research is sort of understanding why splicing defects accumulate in aging and whether that can help us to better understand why certain people age in better health than others, and to eventually sort of help use this knowledge to promote healthier aging trajectories. So I'm sure you've all noticed that some people can age in good health and others will have like multiple chronic diseases. But when you look at sort of their lifestyle and their eating habits, like they're sort of similar. So, I like to give the example of my two grandmas who lived relatively healthy lives. They were fairly active, ate pretty well, but they got dementia or cardiovascular disease in their early 80s and passed away soon after. Whereas my husband's grandmother lived until she was 104 in very good health with similar health habits. And so is it just genetics, or could it be that, my husband's grandma got better sun because she was in Texas? So essentially all of the genetics and environmental exposures that we have throughout our lives could influence how we age. And this could be accomplished in part by inducing or preventing some splicing changes. So one of the research directions that we're pursuing is related to physical activity. We all know that physical activity is beneficial to age, to have healthy aging. But we, at the molecular level, we don't necessarily know how these benefits occur. And so we're testing whether some of the benefits of physical activity are that it prevents the splicing errors that happen usually with aging to maintain or prevent diseases longer.
So by understanding, because from what I hear, is very beneficial for society, basically to understand that the scientific community better understands RNA and how it can be used to in a medical field, and so it basically could allow, once the code is cracked, it could allow us to live not necessarily longer but healthier.
It could. That's, where sort of we're taking our research. And I want to specify that we're really, we're not trying to make people live longer. We're really focused on health span. And so that's how long people can live with a good quality of life. And that can mean different things for different people, but it could mean, staying in their home, living independently, or being free of chronic pain, that sort of things. And so right now, we're very much into the fundamental research at the level of molecules of RNA, and it'll take time for those eventual discoveries to become like applications that will have a direct impact on the quality of life of older adults. But that's the end goal.
Because you also mentioned chronic diseases that could also help. So basically, because you mentioned, for example, earlier you mentioned cancer, but chronic diseases, I guess, is pretty much the same basics, that the body is not sending the message that it should be sending. So does the research could also get to a point where those chronic diseases could be maybe solved or cured down the line?
Yeah, there's a lot of research in that direction, both at the RNA level and in sort of other aspects of these diseases. But, if we take an example of Alzheimer's disease, there are documented splicing defects that happen pretty early on in the development of the disease. And this is not my research, but some other groups have shown that these splicing defects might even happen before the first symptoms start. And so if we could identify those early, even if the therapies that we come up with have nothing to do with RNA, the RNA changes that we see could help us, for example, diagnose the disease earlier. And this is what we call biomarkers. And so those are molecular signatures, sort of, so to speak, that can help us identify diseases early on or before they develop.
To be more into proactive medicine also in the cure approach. Well,
Right, like
not necessarily
prevention,
cure, but also like just treatments. So you because you spoke about your research, so you have a lab, you're part of a lab, you're part of a team at a lab. So what discoveries or scientific advances have you made so far through your research or RNA? Things that you might be allowed to share with us. They're your lab, or part of your research.
Yeah, so my group is still fairly young. I started here in Sherbrooke only less than 3 years ago. So and scientific discoveries take a lot of time. So we have a few things that are sort of ongoing, but not necessarily at the stage of sharing them yet. So I can tell you a little bit more about the discoveries that I made with some of my colleagues when I was a postdoctoral fellow at Harvard. And so I mentioned that splicing, so the removal of that complex code from sentences, is really important. In each gene, so in each paragraph, there are many of those sections that must be removed, and we call them introns. Now, for a long time, splicing was studied at the level of only one intron at a time because we didn't have the technology to read RNAs from one end to the other. So for a while, the way that we read RNAs was by chopping them up in smaller bits and then reading each bit individually. But since about 2018, there's a really cool new technology called nanopore sequencing that allows to read RNAs completely, so from one end to another. And while my group didn't develop this technology, with some of my colleagues at Harvard, we were among the first to use it to study splicing. And we used it to look at the order in which introns are removed from a RNA. And so if I break this down, we know that there's many introns, so many of those sections of complex code that need to be removed. And we might think that this happens, from the first one to the last. But what we found is that that's not the case. There's a specific order, but it could be that, the third intron is removed first, followed by the first, and then the fifth, etc. And this specific order seems to be really important to prevent splicing errors. So if that order is messed up, it will lead to errors that can potentially lead to diseases. So just to make an analogy, it's as if you were building a house. Usually you'll start by finishing the kitchen before you move on to finishing the basement, right? Because you need the kitchen to be functional earlier. So this is sort of a similar thing where even though, you could start with the basement because it's the first floor, you're not going to. Well, in splicing, it seems to be similar where there's a specific order, but it doesn't necessarily go from the first to the last. And as I mentioned, this seems to be important in order to prevent splicing errors.
So when you say cutting the RNA, I'm pretty sure you don't do this in your kitchen, right? Because the RNA, you work at the microbiological level?
I would say the molecular level. So yeah. So it's essentially, it's not something that we can see with a naked eye. We have a lot of tools and technologies that allows us to visualize RNA. But oftentimes, to someone who is just looking into our lab, it might seem like we're just mixing a bunch of small quantities of liquids together. And what I should mention too is that a lot of what we do is actually at the computer, because the methods that we use to read RNA generates like loads of data. And so we spend at least 50% of our time doing what we call bioinformatics, where we're actually using computer science to study our biological results.
So what you said, there's a few interesting things that you said. So you say it's important to understand the sequencing and how the sequencing affects the result, right? Because you say it's not necessarily step 1, 2, 3. 3 might actually be step 1. So that's important because you said prior to 2018, the technology didn't necessarily exist to get the whole picture. So we're studying pieces by pieces and maybe missing the component of how those things interact among each other. The other thing that you mentioned is interesting is you mentioned 2018. So it's prior to COVID-19. Yes. So that's an interesting thing because we're going to have to tackle this. So some people think one of the reasons that RNA gets a bad reputation online, besides the disinformation and the anti-science movement, is a lot of folks seems to think that the mRNA, so messenger RNA, that helped to develop the vaccine, one of the vaccine for COVID, was kind of out of nowhere, like 9 months prior, and people were, one of the things that fed into the worries of the population is that, do we really know what it is? And now you're mentioning 2018, which is prior to 2019, prior to even 2020 or 2021 when the vaccine was first launched. And so when did we start? We're going to talk about that later on, but when did we start actually learning about RNA and studying RNA? Where does it come from for the scientific community to be interested in the topic?
Yeah. I mean, RNA has been part of scientific research, before we even knew what RNA was. The molecule that we would later know as RNA has been studied since like the 1860s. But even beyond that, there are theories that are discussed in the scientific literature, where RNA might actually have been the first molecule that was appeared during evolution, where once upon a time, there might have been a world that was made up only of RNA. There were no DNA and no proteins. And so this is called like the RNA world theory. And part of why scientists think that this is the case is that DNA and protein need each other. And so it's a bit of a chicken or egg paradox where you don't know which one appeared first. But RNA, I talked about it as a message, but it can also be the worker or the machine, the way that protein is. And so we talked about messenger RNA, but there's also many RNAs, including in humans, that are called non-coding RNA. And those are not merely messages, but they also accomplish actions. So they're also machines. And so because of that property of RNA to be message and machine, it's thought that once upon a time, that's how organisms lived, was purely with RNA. And then eventually RNA developed how to make proteins through evolution, and the mode of storing information in DNA followed eventually. And this is in part because DNA is a lot less fragile than RNA. But research in RNA biology has been around, for over 100 years, and perhaps more in higher concentration since the early 60s. And so since 1959, there's about 50 Nobel Prizes in medicine or chemistry that are somewhat related to RNA. Messenger RNA was discovered in the beginning of the 1960s and led to a Nobel Prize in 1965. And then splicing, which is the process that I was telling you about, was discovered in 1977 and led to a Nobel Prize in 1993. So you can see that there's been a lot of research in RNA way before COVID happened.
There's a lot of people listening to us right now who weren't alive. I wasn't alive in those years. In the 1860s, none of us was alive. Well, if you were, you're a vampire, so don't spoil it for people. But so it's been over almost over 150 years, basically, that the scientific community interest has been piqued by RNA, and in the last 80 years, well, 60 years since the 1960s, then it really picked up, and the scientific community gets a better understanding of what is RNA. The code is not cracked completely yet, but we know more and more about it. So yeah, 1960s definitely is a long time prior to the COVID vaccines that we heard so much of. What do you think can explain this?
The code, like the words that are used, so the code that is used to make proteins is really well understood now. And that was part of the discoveries like in the 50s and 60s. What we understand a lot less is why and how that complex code between words is contributing to what we call the expression of the genetic information.
So if we go back to the words, it's basically the printing method has been understood, but which words matter when and how, that part is more complex.
Yeah, or how those words are put together to form the sentences.
If I'm not mistaken, you mentioned earlier the RNA might change also based on the body's function. So I guess even if we crack the code, for example, because I read recently there's I think for pancreas cancer, there's a RNA scientific advance that's pretty new. Maybe if you can tell us about that, but I guess, so if the code is cracked for pancreas cancer, it doesn't mean the code is cracked for all the cancers, because the body has different function. The codes and the words that matter, although we know that those words exist and we understand how they're printed, we won't know which words are important in that specific context. Does that make sense?
Yeah, or like the combination of words, I guess, would be important. Though, yeah. So I guess to talk about that pancreatic cancer advance, we may need to go back a little bit into and briefly explain how mRNA vaccines work, because I don't think we've...
No, we haven't covered the vaccines yet. But if we're there, I mean, yeah, because the vaccines, I think the public scare and the disinformation of the anti-science movement came about RNA mostly came around the time of the COVID vaccine, and it only gotten worse since. But so yeah, I think the COVID thing, or not necessarily the COVID thing, but just the vaccine thing and the mRNA vaccine. I don't want to say technology, but maybe like technology based on the RNA, I think would be a very good starting point, maybe to get where we're at now and how we're going to move forward in science to better understand the applications of RNA.
Yeah. So if we take a step back from what I was talking about, like splicing, and we go back to just the idea that mRNA is a message that contains the information needed to make a protein. So for more traditional vaccines, the ones that were used prior to the mRNA vaccines, those were based on either using an inactive version of a virus or a protein that comes from the virus. And then that would be injected into our arms, and our immune system would recognize it as something that's like a stranger, and would produce a memory of that protein so that the next time it encounters it, let's say if you're infected with the virus, it recognizes it as being bad, and it destroys it. So the idea of the mRNA vaccines is the same, but instead of right away giving the worker the protein, we're instead injecting the message. And then our own cells will use that message to make the protein, and our immune system will mount an immune memory against that protein. one of the reasons why this is like a really valuable advance is that proteins and inactive vaccines or inactive viruses can be really hard to make in large quantities, whereas that it's actually pretty easy to make large amounts of RNA. And so if you inject the message, this can be read into protein by our cells and generate an immune memory so that the next time you're infected with COVID, your cells will sort of recognize that it's something bad and fight against it. So there's sort of two things that branch out from that. The first thing is that there was a lot of work that was done prior to the pandemic to get to the point of being able to develop these mRNA vaccines. So when the media was saying that, it's amazing that the vaccines were developed in 9 months, this was only possible because of decades of research that happened before. So, for example, the two researchers, Katie Carrico and Drew Weissman, who won the Nobel Prize in 2023 for their discovery that contributed to the development of the vaccines, that discovery that got them the Nobel Prize was actually made in 2005. And what they discovered is that if you just inject normal mRNA into our cells, our immune system will see it as a stranger and will destroy it before the message has any chance to be used to make the protein, which is what is really needed for immune memory. So what they figured out is that if you modify the chemistry of the RNA a little bit, so as if you were adding accents to the letters, for example, they're sort of unrecognizable by our immune system. And so this allows the mRNA message to be present in our cells long enough to make the protein that will mount the immune response. And then after that, the RNA is destroyed.
Basically, is it prior to this discovery, is basically our system shut the messenger? We say often don't shoot the messenger, but it's basically our bodies saw a messenger and they just shut it down without ever getting the opportunity to read the message. But thanks to that discovery, basically our body says, oh, there's a message coming in. Let's get the message, read it, and then being able to understand it, because it's basically the instruction manual on how to fight said disease. Is that a good metaphor?
Yeah.
Okay.
Yeah. And so going back to pancreatic cancer, we can use a similar approach to treat certain cancers, where here, instead of being a message that helps our immune system recognize a virus, we're instead using a message that will help our immune system recognize only tumors and kill or destroy those tumors. So the way that it works is there are mRNAs that are specific to tumor cells that are not found in our normal cells. So if we can identify those, the way that it was done for pancreatic cancer, you can generate a vaccine, so an mRNA that you can inject, and then our immune system will learn to recognize that message as being bad. And when it sees it, it will destroy it. But because it's in our tumors, it will destroy the tumors. And this is likely to have a lot less secondary effects than, for example, chemotherapy, where normal cells can also be affected by the chemical that is used to kill the cancer cells.
So it would allow to have way more targeted approach for specific kind of cancer. We're talking about the pancreatic cancer now because that's the one that have great scientific advances. I'm pretty sure many labs are working on other kind of diseases or labs, but it basically shows for those of us, for those our listeners, it shows that RNA is actually a good thing. It's a good thing to understand. It's a good thing for us. It's a good thing for the scientific community to work a better understanding RNA, so that then it can benefit society as a whole, which science does across the board, but we're more focused about RNA today. So what might be the dream, like moving forward? Like if we best case scenario, let's go and dream about unicorns a little bit. What can happen if not we, because I'm not part of the scientific community, but if the scientific community really cracks the codes and understands the whole thing about RNA, what would be the dream of if RNA is completely mastered, which we're far from, but we're moving slow and steady wins the race. So what it might look like, or what's the objective of the scientific community for that?
Yeah, I mean, I think ultimately it's a big component of what we're calling personalized medicine. So, we talked about a vaccine for pancreatic cancer, but researchers, not my group, but others, are working on personalized cancer vaccines, where instead of being against like a certain type of cancer, it would be sort of as considering that each person's cancer is a little bit different from other people's. And so you could take a biopsy from person X's cancer, read out all the RNA and DNA that is in that cancer, and find specific messages or genes that are specific to the cancer relative to that person's normal cells, and then design a vaccine or an mRNA message that can be used to target specifically that tumor. That's really powerful because we know that each cancer from one person to another can be slightly different. And so if we're able to tailor the therapies, that could mean like much better outcomes for cancer patients. And so that's, in development. There are academic labs and companies working on that. And so I think it's like a medium-term thing that will be used relatively soon. But we could potentially expand that to other diseases, like, for example, I was talking about Alzheimer's disease. Can we correct those splicing errors that we see in the disease? And splicing doesn't really have anything to do with the mRNA vaccines. It's a completely different thing. But there's also technologies that allow us, or therapies that can allow us to modify splicing errors. And so those are used in the context of some rare genetic disorders right now, specifically muscular dystrophies. But they could be really expanded to a lot of diseases, so that if we find the specific splicing error that is a cause of the disease and not just like sort of a passenger, we can then treat it.
That's one of the many reasons why I love science, because it helps us understand the world around us. And also, speaking of RNA, it's the world inside us, to be able to correct some dysfunction and to allow us to have better and healthier life. Because chronic diseases are not fun for the person living through them. So if we can find solution, even cancer, I won't say the word, but the common saying online is F cancer. So if we can find a solution to eradicate it, I think we should definitely finance that more. Moving on to my next question, I have to ask that question because I see this a lot, and a lot of people seem to be afraid of RNA because it does sound like DNA. And there's some disinformation that says, RNA is there to modify DNA. Your vaccine will modify your DNA. And what can you tell us about that? Although they sound the same, but I'm pretty sure they're still different. So what can you tell? I'll let you, the expert, tell us what we can understand from RNA and DNA.
Yes, so they sound similar, and they are similar molecules, but they have very different properties. So DNA is actually an acronym that stands for deoxyribonucleic acid, and RNA is just ribonucleic acid. So there's that deoxy that is present in DNA that is not present in RNA. And without going into details, this refers to a chemical difference between the two molecules that actually makes them very different. So DNA, for example, is what we call double-stranded. And so it's like two molecules of DNA that are entwined together at all times, whereas RNA is usually sort of one molecule on its own. So that makes DNA much more stable than RNA. DNA has to last a really long time. If you modify it, it's going to stick around. Whereas RNA, as we were talking about, is much more fragile, and it's very transitory or transient, sorry. It's actually will be made, last a few hours, and then be destroyed, replaced by another, and so on and so forth. And so even if you modify RNA, it's not going to stick around for very long. Now, let's go back to the beginning where we were saying that DNA is in the nucleus. And so it's as if it's imprisoned in a,
It's a fortified fortress. From what I understood from our previous conversation, is DNA is like inside a very secured fortress inside of the cell.
Right.
With a lot of security and guards, and it's like it's no entry.
Yeah, exactly. So I guess the word I was looking for is like a vault or something.
Okay.
It's sort of really well protected by this sort of fence that's around it. So RNAs, the ones that are in our cells, are created within the nucleus, so within that fortress, and then they exit to bring the messages outside the fortress. But the opposite is not true. So RNAs, or at least messenger RNAs, don't usually make the way back to the nucleus. And especially the RNAs that are present in vaccines, they get injected into the cells, but they're not going to go into the fortress. And so they can't really modify DNA. And so there are some different applications where RNA can contribute to modifying DNA, but it's really specific types of RNAs that are different from vaccines and that are not present in our cells, and they must act with a very specific protein. So we call that the CRISPR-Cas9 system, for example. And it's only if the RNA is there and the DNA is there that, the RNA is there and the protein is there, that DNA modification can happen. But it's very specific cases, and all of those components have to be together.
And it's not possible in vaccines, just to clarify for our listeners.
No, because that specific RNA or that specific protein are not present in vaccines.
So if you want to change your eye color, you need contact lenses. And if you want to change your hair, you need a hair dye. It's not, the vaccine won't change your DNA or how you look. So I think it's important to clarify for people, because as we mentioned earlier with your previous answers, RNA application have been studied since over 100 years. So it's not something we might hear about it more now, because one of the reasons why I launched this podcast is we don't hear much about science in the public domain, right? And in easily available newspaper, there's not, like, the scientific message doesn't get around much, very sadly. So I try to solve that with the podcast. But RNA has became very mainstream because of the vaccine of COVID, and COVID was definitely talked about every day worldwide. So people were getting knowledge of the existence of RNA vaccines, but the scientific community have been working on that for hundreds of years. Well, not hundreds, but over 100 years. So they know their way around it more than people might think, that it's out of the blue and 6 months later we get a vaccine. And I think, because that's one of the things I think it's sad from the message that people got, like the general population got the wrong message, I think, for the COVID vaccines, because the message that should have been understood is that when you invest in science, because government were like, we're going to do whatever, like, tell us what you need, we're going to give it to you because we need to get out of this pandemic. I think that's a message that the population should have gotten. Basically, is if you finance, if you give the tool, if you help science will help you back.
Yeah, because, and if we, in the context of mRNA vaccines, for example, people were already working on those for other viruses. And, had been working on the applications for over a decade, and I would say almost 20 years. But when COVID-19 happened, all of this money and all these resources were directed to a single goal, which was to produce a vaccine. And that really rarely happens in the scientific community, where, every area of research is generally underfunded. And so that it's really the concentration of resources that was building upon previous decades of research that allowed the rapid development of those vaccines. But if there hadn't been all that knowledge that was already acquired, such as like how to modify the RNA with like those accents to protect them from the immune system that we were talking about earlier, like that wouldn't have been possible in those 9 months. And so if we go back to the message, I think the media meant well in that they really wanted to highlight how amazing it was that we came to this discovery in such a short amount of time. And it really was. And it was because of the effort of, thousands of people. But the part that was missing from the messaging is that it was possible because we were building upon knowledge that the community already had.
That's interesting, because I think the research might be slow to so most of the time we see the conclusion of the process as a society. Oh, there's a new virus, or there's a new medication available, or there's a new. But how much work is there behind it before the general public, our listeners, can actually get access, for example, to a new vaccine, a new medication, or something, a new discovery in science? What did the scientific community did beforehand to get to that point?
Yeah, I mean, it's generally years, even decades. And sometimes, the things or the discoveries that will help us develop a specific medication were not necessarily meant to be used for that medication in the beginning. It's, what we call fundamental research. So where we're studying a biological process rather than a specific disease. And a lot of researchers in Quebec, in Canada, across the world do this type of fundamental research. So in my case, I'm studying splicing, but it's not always in a health context. Well, a lot of those fundamental research projects have led eventually to medications or therapies, cancer drugs, that sort of thing. But the governments tend to be more willing to fund more medical or clinical research because it seems to give more rapid results, but it's only because it relies on all of those fundamental discoveries that were done years prior. And so essentially, like if you're meeting someone or a researcher and you ask them like sort of what is their research on, like generally the general public means like which disease is your research on, but it doesn't have to be on a disease. It can be on a biological process, and eventually it might be applicable to a lot of different diseases.
That's interesting, because you say, for sure, government, it's more flashy when governments say we invested, I don't know, X amount of millions in this technology, and boom, we have a new medication or whatever. But the fact of the matter is that what allowed it to happen, if I may air quote, that quickly, is the many, many years of research behind it that helped to understand the biological process, as you mentioned, that allowed for that medication to even exist. So I think it's important to also, maybe as a population, to be more sensitive on how important it is to finance that fundamental research, because we're going to talk about it in a few moments, but there's a lot of cuts in science right now, which is, as a society, that's not the direction we should be taking, because as you said, it also takes time, right, to achieve those success stories. So we need the resources to invest that time for the benefit of society. But which brings me maybe to my next question, because we are sadly in a time where the anti-science movement is getting steam a lot, like it's more and more popular, sadly. And one of the objectives of the podcast is to address misinformation. And so what are some common misconceptions in your field that you'd like to clarify or correct that we might not have addressed yet in the episode?
I think we've covered them mostly so far. So, that RNA can't modify DNA, and that the vaccines are unsafe because they were developed so rapidly. That's, I hope that I've been able to illustrate that that wasn't the case at all. These are the main aspects of misinformation that we've been hearing about RNA mostly since COVID. It's not there, I don't think that other aspects of RNA research have been as much a target of misinformation.
So you did a good recap, because I wanted to do a recap also about that. But if we recap from the beginning, so for our audience, so RNA has been discovered, if I may, like in the 1860s. So it's been a while.
Yeah. Well, I mean, what would become known as RNA was
what would
first
become
discovered.
known in the 1860s. The scientific community has been working on it for decades. The first Nobel Prize related to RNA research is in 1965, which most of us, I wasn't alive, definitely. And so it's been a long time. It's not out of the blue. RNA, the mRNA doesn't changes our DNA, or the vaccines won't modify your DNA. It's important to invest in science, fundamental research, because that's basically the hard work that allows maybe to bring to the more fancy work, if I may, more fancy discoveries. And understanding RNA can really unlock the key to solve and to develop treatment for chronic diseases. And also, I like the expression that you use, the healthy aging, healthy years, health span. So it's not just how many years. It's not necessarily living longer, but it's living better and having a better quality of life, which we all aspire to have. Basically, everyone listening, we all want to hopefully live long, happy, and healthy as much as possible, which RNA can help us with the discoveries that the scientific community who study that subject do. Is that a good recap? Did I miss something important?
No, I think that's
good. So, taking inspiration from a scientific paper, the structure of a scientific paper. If you want to know what a scientific paper looks like, I invite you to listen to the episode 1, if you haven't, which explains the scientific method. But taking inspiration from the conclusion of a scientific article that opens doors for future research in the field. What can we expect from you, Karine, as a researcher, your team, or in your field?
Yeah, so I mean, I hope that in the next 10 to 20 years, my team's work will allow us to better understand how RNA contributes to different aging trajectories, and more importantly, to design some molecular strategies so that we can allow older adults to age healthier and to have better quality of life. So that's one point. The other one, which I didn't talk as much, but genetic disorders are generally caused by spelling errors in our DNA. So that's what we call mutations. And those mutations can also cause splicing defects. And so a big aspect of the work in my lab is to understand why specific spelling errors cause specific splicing defects. And I hope that this will allow us to eventually develop more targeted treatments for specific genetic disorders. And we're working on different types of muscular dystrophies. And then one of my areas of particular interest, which a few members of my team are working on, are those non-coding RNAs. So remember how I talked about some RNAs that are not messenger RNAs, but they can act themselves as machines, so they can do the work themselves. There's actually a ton of non-coding RNAs in our cells, but they've been really understudied compared to messenger RNAs. And we're just starting to appreciate how much they may contribute contribute to genetic disorders, cancers, neurodegenerative disorders. And so I think it's going to be really fascinating to continue to discover how they can contribute to modulating our health and whether they can be targeted to improve our health.
So basically, so you say the purpose is to understand the misspelling error, like the grammar mistakes maybe there is in DNA. So would solving that issue could actually teach our system to basically spell correctly and so tackle that bodily dysfunction that come from chronic diseases?
Yeah, that's essentially what's already being done in a few genetic disorders where they've figured out sort of the reason behind, or they figured out the consequences of the spelling errors on the message. So the spelling errors lead to the words being put together not quite the right way. And what those therapeutic approaches do now is to use small molecules that will bind to the RNA before splicing happens and will allow splicing to change so that we can again create a sentence that makes sense. And so this is done for a very small number of genetic disorders right now. And part of the reason why is because we don't know yet which other genetic diseases can be targeted in that way, because we don't fully understand the consequences of the spelling errors. So if we can understand the relationship between the spelling errors and how the words are put together and how that causes symptoms, then we can try to fix it.
Well, I know I say it often, but I love science. I love science so much. Yeah, lucky there are people much smarter than me, because otherwise the society we would be in a tight spot, but that's good to know. So the floor is yours. I like to give the closing comments to my guests. So what would you like to tell our audience before we go for the outro?
Yeah, I think I'd like to go back to what you briefly alluded to about how, there's a bit of attacks on science, maybe not so much in Quebec and Canada, but certainly south of the border, including against RNA research. And, scientists are often described as being sort of like the minions of pharmaceutical companies. And what we do is really just for pharmaceutical companies to get richer. And I'd like to really dispel that idea, because most scientists are just passionate people. They're passionate about their topic of research, and they're not motivated by money at all. We really do our work because we want to contribute to improving health and quality of life of people in our communities. And, it's really like university researcher and professor is not a job that you do for money. It's a job that is very demanding. It requires 10 to 15 years of studies and postdoctoral training. It's a really large workload. Most of us work often on weekends and on evenings, and we're constantly applying for more research funding. And so I don't want to make it seem like a bad career, because I love my job. Because all of those challenges are worth it, so that we can have the privilege to pursue our passion and to be on sort of that first to be the first witnesses of really fascinating discoveries, and to contribute to training the next generation of scientists. And so related to that, I want to emphasize that research scientists are not sort of working alone in their labs, as we're sometimes pictured like in movies or TV series. It's usually a really big teamwork. So the success of my research and that of many colleagues is really because of my entire team, which is composed of graduate students, undergraduate students, and research professionals. And so when you see like a Nobel Prize or another type of discovery, it's extremely likely that there's an entire team that was behind that discovery, beyond just the one or two people that are getting recognized. And I think it's important to remember that.
Well, Very good closing comments. And yes, definitely, that's one of the things I think it's important that people really realize, our listeners, is that especially in academics, you don't do research for the money because you're in the wrong field. Definitely, if you're there for the money, there's something you misunderstood. But people are there for the common goal. So all those nights and those evenings and those weekends that are worked for, they're worked for the greater good, literally, of society as a whole. So, well, thank you very much. I'll put the relevant links in the episode description. Also, for our listeners who want to follow us on the other platforms and go see your research website, your research lab's website. So, thank you very much, Karine, for your time and your expertise, which allowed us to learn stress-free about RNA and its relevance to science and its many benefits for science. To my listener, thank you very much for tuning in. See you soon. I love you all.
Thank you so much for listening to our whole episode. I hope you learned a lot and had as much fun as we did. If you want notifications for new episodes, you can subscribe to my channel on YouTube or on your favorite podcast streaming platform. You can also like, comment, share with your friends and family on top of subscribing. It's a great way to support the podcast free of charge, allowing me to bring you even more subjects to learn stress-free about. You may also join my Patreon and follow me on social medias to get news and teasers for upcoming episodes. All relevant links are in the description below. I'm curious, what have you learned in this episode, or what surprised you? Leave us a comment and let's keep the conversation going. To conclude, a huge thank you to you, the listeners, and to our guests. You all make that podcast possible. See you soon for another episode of Learn Stress Free. Until then, keep learning. I'm your host, Jeff Rodrigue, signing out, wishing you the best. I love you all.